Hi everyone, thank you so much for coming and welcome. So I wanted to introduce our speaker for today, Mark Leake from the University of York, who is the anniversary professor for biological physics. That was 50 years anniversary of University of York when you...
It was, yeah.
Yeah, well, 10 years ago, something like that.
Yeah.
And then he spent his career at various places, I think Cambridge for a master's and King's College for PhD and then Oxford for postdoc. That's actually where we met. When I visited Judith Armitage, she worked on bacterial chemotaxis and yeah, you were a postdoc. I just remember quite vividly still going after the seminar to the pub, and it's always fun to go to the pub and talk some science with Mark after the talk. Can highly recommend. And so on and so on. He went on, I think he spent also some time in Heidelberg. So he went on to do have a fantastic career. So he developed very important instruments or super resolution, single molecule stuff in biophysics and imaging. Worked on various interesting topics, DNA replication, molecular motor, and so on. Really good stuff. And and then he also contributed quite a bit to the community in biological physics. So he's currently chair of the biological physics group of the Institute of Physics, IOP. It's a bit like APS or so on in the US. And what else? I mean, he's running the Physics of Life Networks, the UKI-funded network, PolNet 3, which was recently renewed, just very recently. So let's welcome Mark.
Thanks so much, Robert, for those warm words. And it's really great to be here, actually. And thanks, Will, for organising the day. I mean, so much wonderful activity here at the interface between the physical and life sciences. It's super to see this sort of emergence of critical mass right here. And one of the beauties, I think, of the system that I describe is that I think it's, for me, it really demonstrates the value of physical science approaches to gaining real sort of biological insight. And there's very much a sort of close interplay between the life sciences and the physical sciences, and between experimentation and theory and computation. And so, a few years back, maybe it was in the same pub, I don't know, I had a bit of an epiphany that the way I wanted to do this sort of science is really, at that sort of very sort of integrationist level. So instead of trying to have everything kind of in-house as a research head, I really wanted to just reach out to the people who knew what they were doing, and to have this sort of strategy, this framework of integrating different tiers of expertise to solve big problems. And I think that this is the thing about biology, my understanding of it, is that to really get some level of insight into biology requires these multiple tiers of expertise. So, it really isn't just about having a physicist and a biologist talk to each other. You've got to think much, much bigger than that. And you've got to buy people in at the very earliest stages of a project. So, you can't afford to do like a whole range of experiments and then sequester in some theorist at the end and hope that they'll have some wonderful modelling insight. You really got to embed that right at the early stages, because what you'll find is that the nature of the questions change. That's the thing. You get this wonderful reframing of research ideas, hypotheses, provided you give the right amount of time to enable that reframing to work. As you can see, quite a lot of people contributed to this, and this is the multiple tiers of expertise. And the focus of this talk, the system, the biological system, is this remarkable liquid droplet structure called the bacterial agrosome. And I'm sure many of you are aware that liquid phase separation in biology is quite a popular thing. in our research community at the moment. There's a danger that, maybe it's a bit of a fad or fashion, but the ubiquity of liquid droplets in cellular systems demonstrates clearly how useful they are. And limitations as to our insights into these features have been at two levels. One, it's at a theory and computational level, and two, it's at the level of technology, because as I'll show you, these droplet features really are very metastable. They're very small and they're very transient, and so this makes it very challenging to try to follow them with conventional bioimaging tools. And so, part of this work has motivated technology development to basically have imaging systems that are, more sensitive and faster. You see this movie again in the talk. Essentially, this is a sort of made-up E. coli cell. It's a rectangle. And you'll see that the simulation qualitatively recapitulates the features of this liquid droplet formation, and that you get these two largely polar localized liquid droplet structures. Now, I'll divide this talk into describing the discovery of this bacterial agrosome feature. It largely contains hundreds of proteins. There's no phospholipid bilayer, but we would rightly call it a subcellular organelle, but in bacteria. I'll talk about how we have characterized its liquid nature and dependence on cell stress. Then there'll be a bit more of this type of modeling and discrete Monte Carlo simulations to try to quantitatively explain the physics behind these droplets. I'll then show more recent work how RNA is a component, as well as proteins, of this droplet system, and how the RNA has got a length dependence. So essentially, longer RNA is more likely to be captured by these droplets. I'll show you how we know now that the enzymes that are responsible for degradation of RNA in the cytosol are excluded from these agrosomes, and why we think probably the key biological role of these agrosomes is actually to protect RNA transcripts from degradation during stress. And then if I've got a few minutes at the end, I'll talk to you about some opportunities in the UK community, about engaging in UK condensates network, and also I'll talk to you a little bit about the wider sort of physics of life activities that Robert alluded to at the top. Okay, so these agrosome features, they basically form in bacterial cells. This is model E. coli system in response to a whole range of different stress factors. So we can throw antibiotics at the cells, we can heat them, we can give them a cold shock, we can add toxins, and we can starve the cells, and we see these little sort of black foci that you just squint, you just start seeing them at the sort of poles of the cell. This is just normal bright field microscopy. Now, if you know a little bit about microscopy, that will tell you that, well, to see these as dark foci in these images means that the refractive index must be changing by a reasonable amount, so that there really is a lot of stuff inside these droplets. So, one thing is, they seem to be quite tightly packed. And what we can do, actually, subsequently, we can do proteomics on these extracted agrosome features, and we can work out the cocktail of proteins present in the agrosomes and look for candidate so-called biomarkers, and then put a fluorescent tag on one of those, and then we get these much higher contrast images that you see in fluorescence. Now, I want you to draw your attention to these movies at the bottom, maybe focus on this one here. You can see these the dark foci in this E. coli cell. Basically, at the start of the movie, the cell is surrounded by antibiotics, and then what happens is that we flush out the antibiotics in a normal growth media or minimal media in this case for the imaging. And you'll see that the dark foci disappear when we remove that stress factor, and then you'll see something interesting happen to the cell. This is if the movie runs. If only I had your MacBook Pro. But let's see. Oh, he's running. Great. So look, dark foci, and then arrow goes, foci disappear, and look, the cell magically starts dividing. Essentially, what has happened there is that cell, when it's subject to the antibiotic stress, has gone into a quiescent, like hibernation state. It's the so-called persister cell. And so it sort of shuts down, almost all of the metabolic processes, just kind of sits there. But then you remove the stress, the acrosomes disappear, and then obviously these metabolic processes get switched on because the cell starts dividing again. And so you'll see that coming to the conclusion very early on, essentially we think that these acrosomes basically contain all the goodies in the cell required for it to resuscitate after you have removed the stress factors. So the talk is sort of leading up to that. So I mentioned preliminary proteomics work. We spent a while working out how to actually extract these acrosomes from cells. Essentially, you can crack open the cells, the cell lysate, you spin them down because there's liquidy globs, and then we optimize pull-down assays, and you can purify and do gel extraction, and you can purify these acrosome components. There's clearly a bit of fusion en route because the size of these extracted acrosomes in vitro is, effective diameter a micron or two, whereas inside a single cell it's more like half a micron. So that gives you a clue as to this metastable morphology of these droplets. What it means is that if you can extract them, you can do a lot of biochemical testing, biophysical testing outside of the cell. So this is mass spectrometry data. This is what the sort of bread and butter tool for this type of proteomics analysis. Essentially what this means is that we can pull out the individual proteins that are actually present in a population of thousands of these acrosomes. And without guiding you through each one, what I can say is that several hundred different proteins are present inside acrosomes, but the mixture of these proteins changes depending upon the cell stress and depending upon the time after which you have subjected that cell to stress. So, for example, at early stages you'll see proteins associated with other protein production and electron transfer reactions, and then in later stages of cell stress it's also DNA replication and repair. So that gives you a clue that you've got quite a dynamic system as well. It's responsive, agile towards time-dependent stress changes. So moving this onwards, this is all sort of the expertise in my lab. The core expertise is really sort of biophysical quantification at a very precise single molecule level. And so what we thought would be valuable is can we put a number on how many of those proteins are actually present inside these acrosomes? Because if you can, that number can feed into models, hopefully, that will emerge, and then if you can do that, you've got a robust predictive framework that tells us something important. And so using the initial proteomics data, we did various screens and we came up with a handful of candidate biomarkers. And you can see this basically shows the fluorescence intensity of these acrosomes, the three different candidate biomarkers as a function of time after the start of the cell stress. And so you can see the dynamics of incorporation of different proteins is different as a spectrum. So we selected one, which is a protein called HSLU, which is a heat shock protein because it's quite an early responder to It's made the experiments a bit easier to do. And then we use that subsequently in most of the experiments, that sort of go-to protein to tell us what's happening in the agrosome. So one of the first things we did was apply a workhorse technology in my lab called slim field imaging. This is basically where you underfill the back aperture, the high numerical aperture objective lens, and it's a trick that's been used by the fluorescence correlation spectroscopy community for ages, but they never applied it to actual imaging. It means that there's a reciprocal relationship between the width of the beam that goes in and the width of the confocal volume at the level of the sample. So what that means is that you can change the width to be just a bit larger than the single cell. So it's just a very cheap way to get a lot of light into the sample, and in doing that you can carve up the finite photon budget of these rather poor fluorophores of fluorescent proteins into millisecond chunks. Why that's important is because that sort of is the diffusion time scale in the cell. Robert?
Yeah, I was wondering now, so that's diffraction limited, yeah? So what you're seeing down there, these circular blobs,
Yeah.
that is not the actual size, right? It's limited by the wavelength, which is a few hundred nanometers, basically.
That's right, yes.
So in the previous slide, you had lots of proteins in these aggregates,
Yeah.
like several hundreds or thousands even.
Yeah.
So I mean, how big are they actually, these aggregates? I'm a bit confused now. I thought they're
Sure.
generally smaller.
Sure. So what we can do are basically more sort of super resolution type tracking techniques, in which even though you're sort of limited, you might sort of get down to stochastic activation or bleaching down to the last fluorescent protein, which you can visualize in the agrosome. Then you can just use Gaussian fitting to pinpoint the intensity centroid of that function. That gives you a much more refined estimate as to where it is, maybe a few tens of nanometers instead of 300 nanometers. And then by tracking that, that tells you something about the dimensions of the agrosome, because then you can do mean squared displacement analysis. What you'll see is that you get an initial sort of linear bit, and then it starts to plateau, and the plateau is roughly where the edges of the droplet actually are. And so there are other super resolution techniques that show you like structural elimination microscopy. But to answer your question, what we can actually visualize, the smallest structures, a few tens of nanometers in diameter, and it sort of extends up to a few low hundreds of nanometers. The cell itself is a micron wide. But you see from the subsequent modelling that I'll show that it's classical nucleation. So you'll start off with just literally a handful of these proteins coming together, but it's then determined by the sensitivity of the detection system actually when you start actually being able to visualise them. Okay, so we can track them, right? And we can turn the brightness of the light that we see into a metric of stoichiometry, how many of those fluorescent proteins are there, because we know what the brightness of a single GFP or whatever fluorescent protein molecule is, because it photobleaches in a stepwise manner and you can measure the size of that step.
Just to follow up
Sure.
for a second, so these droplets occur during stressful times, right? And the cell is dormant.
That's
so
right.
they ripeness, right? So they would grow bigger and bigger essentially with time, or?
That is the right question to ask. The weird thing that happens is that you do get this ripening, but instead of keeping on growing, as you might expect in sort of classical sort of phase transition behaviour, where you basically use up all of the material, that in general doesn't happen. You have these preferred length scales, and why that happens. Well, this is an open question in the whole of the the biomolecular condensate community. What are the features that limit the length scale of these systems? So with this tracking, we can turn that brightness into an estimate of number of protein molecules, and so for example with this HSL mu, you can see it's a few starts in the early stages here with a few tens of these molecules, and then goes up to several hundred. But you've got to remember that you've also got several hundred of these other proteins. So it is quite a gloopy, tightly packed environment.
Also very variable, right?
Huge.
The rate thereabouts of that.
Yeah. Absolutely, absolutely. But don't forget the tagline of all of this is that these are really, really very metastable structures, and so you've got a whole range of fusion events in particular that occur. So different spectrum of behaviours in terms of the stoichiometry that you see and the effect of the apparent diffusion coefficient. So we were interested in aspects of liquid-liquid phase separation in general, and some of the early indicators suggested that this was a sort of a good candidate for liquid-liquid phase separation. So, for example, this is an example of structured illumination microscopy, another super resolution technique, and you can kind of see these sort of spherical-ish droplets, but it's not, it's clearly sort of beyond just a sphere. It's it's much more metastable than that. You can see fusion events between different droplets. If you wait long enough, for example, you can see these two spots here become a single droplet in living cells. And you can also see if this movie runs. We can also we can also recapitulate some aspects of the phase separation behaviour in vitro and see the sort of classical kind of fusion behaviour here. So again, these are sort of the typical indicators of liquid-liquid phase separation behaviour. You can also disrupt these droplets using a chemical called 1,6-hexane-diol, and this is sort of your go-to organic chemical. But bear in mind that's 20% of so-called hex, and 20% is absolutely huge. So you're disrupting loads of metabolic processes. So take that with a pinch of salt in and of itself. But in the round, it got lots of indicators of liquid-like behaviour. And similarly, we can do so-called fluorescence recovery after photobleaching, or so-called FRAP. We can do this in two ways. One way is half FRAP. So here we delimit the excitation volume of a high-intensity laser beam to preferentially photobleach just in half of the components of the droplet, and then we can follow the subsequent recovery fluorescence into that bleach bit. That tells us something about the mobility of the components within the droplet. But we can also do so-called whole FRAP, which is here, and when you see recovery of fluorescence of that bleach spot, just about here, that means that you must have turnover as well. So unbleached components are exchanging for the bleached components from the outside, the cytosol, for the components on the inside. So again, obviously a very dynamic system. So as I promised, you see this movie again. This is the one at the top, and this was work of Charlie Schaefer and late Tom McLeish, and basically this is the first attempt using the standard relatively trivial continuum modelling embodied by this Kahn-Hilliard equation, and it recapitulates qualitatively the features of droplet formation. So you can see that they kind of form roughly where you expect them to see them from experimental data. It's relatively easy to simulate. But the timescales are wrong. They're far too fast compared to the experimental observations. And also you can't see individual molecules, and that's a pity, because you can do on the experimental data. So that motivated us to developing an improvement to the simulation strategy. And so this was done again. We have the same sort of naked E. coli cell, which is a 2D rectangle, but now we divide up that rectangle into pixels of 50 nanometer edge length, and we allow components in each pixel to hop stochastically to their nearest neighbours. We have a mean field approximation to approximate the force fields which are contributed from these hundreds of proteins. And then we have in effect, we have a sort of biomarker that hops around this mean field, and we ask the question when it does this hop, are you likely to stick in that pixel and with another biomarker molecule or not? And the probability of that sticking is basically proportional to this optimum factor. And the key parameter here is this delta U, this entropy of interaction. So it's pretty easy if I just sort of run a simulation and see. So you can see, you start off with these really some preliminary nucleation events, and then you start getting these two droplets formed. The red indicates these photobleached components, so we can simulate the frap as well, and we can track all of the molecules in the system to essentially do everything we can do in the experimental system. And the beauty of that is that we can then compare the experimental data with the simulations as a function of the different delta U parameter. And in optimizing, we work out what the delta U parameter is. Is that
Yeah. So just looking at these simulations,
Yeah.
so the bright blue spots, are they the photoactive?
Yeah.
Okay, because they look. There's something else in there that's helping to glue those aggregates together, is it? Because they look relatively diffuse within the aggregates. Is there a dark blue colour in there?
There's no dark blue. All you've got there is you've got water in the system. You've got these biomarker proteins, and then you've got either sort of photobleached or not photobleached, and then you've got energies of interaction embodied by this delta U parameter. So
Okay,
it's very strict answers.
So but then, am I right that the clusters are not very tightly packed?
You're exactly right. Yeah.
Okay.
And in fact, the delta U parameter, this is like your scale for how well glued together they are, is about 2 kT. So it's just above the noise floor of thermal fluctuations of the surrounding water solvent. So yeah, these things form but then break up stochastically all the time. That's how unstable they are. So it's not like a sort of classical subcellular organelle that maybe you're more used to, like in the mitochondria or the nucleus. That's like infinitely rock solid compared to this. This is super fragile, but also it means that it's very responsive to the microenvironment, rapidly responsive to environmental changes in the cell. And this is why we think this is actually useful. And I should say another nuance here. There's no sort of additional free energy that has gone into this system. So if we just run it for long enough, you'll end up with one massive blob, sort of quasi-equilibrium. So really, these two droplets are like snapshots en route to path of equilibrium. But the reason why we don't see them form in live cells is that there's a cell division time, and every 20 minutes or 30 minutes or so in this sort of full rich nutrient media, these cells will divide. We can prolong that by having minimal media to 1 or 2 hours, but it still hits that sort of natural endpoint where the cell then divides. So we then did a genetic screen, and we found certain candidate genes that if you delete them, the cell, for whatever reason, is less likely to form agrosomes. And if you then subject those cells to stress, then basically this is the wild type, and these other colours here are the this yellow and green. They are less likely to survive. So in other words, there's an increase in effective bacterial fitness if you have these agrosomes present. And we can also do a chemical perturbation. There's a buffer called MOX, acts as an bottom right. And if we add MOPS, again, we can see that these cells are less likely to form aggregates. And again, that's rendered in the red bars here. And these cells, compared to the wild type, are less likely to survive. So there's something in these aggregates that is increasing their viability during stress. So it's not just unique to E. coli cells. We looked at a whole range of other so-called gram-negative bacteria, and you can see these dark foci cells which have been subjected to stress. And we're also looking at gram-positives, and we can see some of the dark foci. So we think that the formation of these aggregates has got a sort of ubiquitous nature to it, as in terms of being a sort of generalized stress response in bacteria. So up until that stage, we thought that these aggregates were largely, proteinaceous in nature. But then there are clues from the eukaryotic cell biology community that maybe proteins are just one of, two components, and RNA is, a very common component in so-called stress granules, which is the mammalian cell, eukaryotic cell equivalent of these aggregates. Now, the issue with trying to visualize RNA in live cells is that the go-to technology, which is RNA fish, means that you have to chemically fix the cells. You also have quite large antibodies that then sterically hinder a range of different processes. And so what we wanted to do was develop a better technology that would at least allow us to visualize RNA inside living cells. And what we did was develop or rather modify an approach using so-called aptamer labeling. And here we could genomically encode these aptamer binding sites onto specific mRNA transcripts. The aptamer then specifically binds to a bright organic dye, and we worked out methods to get that organic dye into the cell very efficiently, so that at least 90% of the RNA present in the cell is actually labeled. And what that means, I'll show in a slide's time. You can actually visualize single molecules of RNA in living cells. So this in itself, forget about the whole rest of the talk. The ability to do that is transformative, because we can really just track single molecules of RNA, millisecond time resolution with living cells. Now here you can see that our go-to protein biomarker HSLU and GLP-K-pepper is the name of this particular aptamer. GLP-K is just one of these candidate agrosome proteins, RNA transcripts present in very sort of enriched concentrations within the agrosomes. You can see that upon stressing the cells, that you get qualitatively co-localization between the RNA and the agrosome biomarker. So you think, we thought at that stage that, okay, so RNA seems to be incorporated into the agrosomes as well as protein. And then we could, explore and ask the question, what is there something special about the RNA that's actually going into the agrosomes? And a simple way to look at this is to use something called screen tape analysis. This is a type of gel electrophoresis, but it's quite precise. It gives you a metric of the length distribution of the total RNA present in either the cytosol or in the agrosome component. And you can clearly see that there's a bias towards larger transcripts within the agrosome. And we think that makes sense. This is basically sort of the go-to polymer physics model, fluorine Huggins theory. Basically, without going into the details of the model itself, in essence, there's a non-specific interaction between the negatively charged phosphate backbone of the RNA and the slight positive charge of the soup of protein. So if you have longer transcripts, there's more of a pull to have that protein incorporated into agrosomes. So in other words, there'll be a bias towards longer RNA. We can optimize that, and that agrees quite well with the experimental data. And this is a demonstration that we really can get down to single molecule levels. So for example, we can do a nice clean in vitro assay, put this pepper construct onto a clean surface with RNA, and we can see these individual spots and see a distinct photobleach step. So we can really get down to single molecule precision with mRNA, and do super resolved single particle tracking, and work out things like the effective diffusion coefficient of the RNA transcripts within these agrosomes. And one interesting feature is that there is an age-dependent age-dependent increase in viscosity, and you can kind of see hints of that qualitatively. So if you look at 30-minute time point after the start of the cell stress and the 60-minute time point, you can just about see that the size of the blue fuzzy bits is a bit smaller in 60 minutes, and the size of the pink fuzzy bits is a bit smaller. So in other words, there's a sort of compaction effect. So everything's getting all sort of more scrunched in, and so maybe it's not surprising that you get this increase in local viscosity, and that's manifest in this subsequent decrease in the effective diffusion coefficient. But as you can see, again, a whole range of behaviours here. So it's quite challenging to extract information from these data, but you can see these trends emerging, and the sorts of directions that we're going with this research. I won't show you this, but we're looking at more sort of mechanical probing of the agrosome properties, using things like optical tweezers and AFM force mapping. So using these tools to try to mechanically perturb the morphology of agrosomes and see how the spatial patterning of these different components changes in response to that mechanical perturbation. And that gives us, hopefully some level of insight as to the biology that's happening in terms of what these different components are actually doing. So sort of getting on to the sort of tagline of the story now, which is that if you look at the enzymes that are responsible for the degradation of RNA in cells, they're called RNases. We see that these appear to be excluded from the agrosome. So, for example, you can see one such here, RN, and again, the blue is the protein biomarker HSIU, and you can see that there's not a great deal of overlap between the blue and the pink. So again, you take this with a little bit of a pinch of salt because it's it's diffraction limited microscopy days. But we can be a bit more clever than that. We can do some genetic perturbations on the RNases. So, for example, we can change the surface chemistry. We can change the electrical charge of these proteins, and we find that if we change, for example, aspartate residue to a glutamate on the outside, we can switch the electrical charge so that it goes from a net positive to a net negative. Then, if you remember, I suggested there's a small, the mean electrical charge distribution within these agrosomes is a little bit positive, and so it means that these modified RNases are then drawn in to the agrosomes, and we can actually do the screen tape analysis on the extracted agrosomes, and we can see that the RNA within these agrosomes is then degraded. So in other words, if you exclude the RNases from these agrosomes, you're protecting the RNA from degradation during stress. And this last key slide, it demonstrates that, because had that concept because what you can see here is, well, at the top here we have, again, sort of standard formation of agrosomes in response to stress. You see up to this point here. And then we remove the stress factor, and you can see that these agrosomes disappear, and there's diffuse fluorescence both from the RNA component and from the protein component. Now, if you do the same sort of experiment that you add in antibiotics that either suppress transcription or translation of the RNA that was present inside those protected agrosomes, then what you can then see is evidence that new RNA is then being made in the agrosomes that are dispersed after you remove the stress factor. So in other words, there is this residual amount of RNA that is present and can still be visualized and actually converted into protein. So, we're still ongoing work because we're looking at a whole range of different RNA transcripts. What we really want to do is understand the physical rules of tunability, of selection. How is it that that certain RNA goes into the agrosomes but some RNA doesn't? And it's not actually as simple as this length scale dependence because there are key transcripts that buck the trends. There's one component called MUC-F, which is relatively small. It's one of these so-called condensing type molecules. But even though it's relatively small, it's found in great abundance within these agrosomes. So there's something more nuanced going on about the specificity. And our hope is that if we can understand these physical rules, then maybe we can re-engineer this system and get synthetic agrosomes to sequester recombinant molecules, recombinant RNA. For example, next generation vaccines, gene silencing reagents, because then you've got quite an appealing system in which you can have a bacterial cell that overexpresses a next generation therapeutic, and then you use the agrosome to protect that therapeutic from degradation, and you can extract that agrosome and release the therapeutic later and increase the manufacturing efficiency by 1 to 2 orders of magnitude compared to the sort of state of the art at the moment. So this is how big it could be. But there's a lot of very basic experimental and theoretical biophysics that needs to go on before we're at that stage of being able to tune the system. But that's the hope anyway. So look, I promised you that I would talk a little bit about wider opportunities in the community. A few of us, Chafan included, thought it would be a good idea to try to explore the possibilities of a UK network covering aspects of biomolecular condensates. And for me, the reason why this is a valuable exercise is because I can see that there are sort of 3 fragments of different research communities emerging. There's the sort of chemical engineers who want to make biomimetic systems, the biologists who are interested in what do these droplet-like systems do for my biological questions, and the physicists who are interested in the basic biophysics of liquid phase separation. And these little 3 entities are kind of phase separating themselves. And it could well be that maybe that's okay. Maybe as a national community, we should just organically let that happen and see what happens. But it's at such an early stage that I think it's ripe to have that sort of conversation and to sort of get the community to swap notes and maybe have a sort of think in a different way. Because if you do that, the chances of getting more money into the community, if you lobby with a single voice, is much higher, I think. So I think it's good, and we had a preliminary network meeting in January in Durham, and I think that was successful. There's an email list if you're interested. Just go to Google and put UK Condensate Network, just mail, and you'll find it. You can subscribe, and you get all the stuff. starting a web page, and we'll probably have another meeting maybe in September or so this year. Secondly, there's the Physics of Life in the UK, and I'm also chairing what has been quite a long roadmap consultation exercise. We've basically gone around disseminating the ideas of physics of life to different stakeholders in the UK, trying to gather evidence. You may have seen various community surveys coming away. I hope you responded to some of these. And we're going to present the findings of this roadmap at a discussion meeting, which will be part of the Physics of Life 2025 conference, which is in Harrogate next month. And if this hasn't been on your radar, do check it out. We had our inaugural meeting in 2023, and it was great. We had 500 participants. We had stakeholders from a whole range of different research communities in this very heterogeneous biophysics-y type landscape. From the physicists through to the biologists and engineers and chemists and mathematicians and computer scientists. And it all worked really well. A very sort of good, productive, safe environment. And I think you would like it. So if it hasn't been on your radar, just check it out. Look at the range of talks. Harrogate is a beautiful little town in North Yorkshire. I think you will absolutely love it. And it's a great networking opportunity as well. And, talking about, networking opportunities and why you network, EPSSC gave me a bit of cash to follow on with this work. And so I will be advertising for postdocs in the not too distant future in a whole range of different areas. So the truth is I'm sort of really more interested in enthusiasm than in sort of your core expertise right now. People in my team, basically, they all are very eager to move out of their comfort zones and learn new tricks. Whether it's experimentalists learning how to do computational work, or the other way around, or life scientists trying to work out how to adapt this type of single molecule biophysics technology. The opportunities are all possible, but you've got to be sold on that idea, and it's not for everybody. But if you're remotely interested, just drop me an email, and then we can keep up that conversation. So let me just leave you with a few questions to summarise what I've shown you. I've walked you through the discovery of these weird metastable subcellular organelles called agrosomes in bacteria, and talked to you about their response to cell stress and how we think they're the bacterial equivalent of a stress granule. I've also told you how RNA is recruited and how these enzymes that degrade RNA are excluded, and that we think that these agrosomes serve a primary role to protect RNA during stress. I didn't really follow this up, but at the start, if you remember, I showed you this movie of this cell recovering after the removal of stress. I called it a persister cell. And you see that the cells that are the most successful persisters are the ones that have the highest prevalence of agrosomes. And persister cells are the ones that go on ultimately to be superbugs. So there's a whole sort of link to antimicrobial resistance, which is interesting in and of itself in terms of the physics and the biology. But also, if you think about it, it presents maybe an alternative candidate for a therapeutic target in this liquid droplet of the agrosome. And I'll stop.